the purpose of the workshop is to take the training to another level. So they've been involved with doing species collections and field identifications, and now we're training them in methods of higher level of identifications to the point of describing new species, which is quite a complicated task. So it involves genetic analyses, photography, morphological measurements and analyses, and then through to the writing of scientific papers. So it's a whole nother level of work.
A few years ago, the EAF-Nansen Programme has a research vessel, the Dr. Fridtjof Nansen, and so we do surveys at sea, and we actually collect specimens during these surveys, and we needed to have an institute where these specimens could be preserved. And so I contacted SAIAB to see if they were interested in keeping these collections from our surveys, and that's where all the let's say collaboration started with SAIAB from there.
It's difficult to find a place where you can do this kind of workshop that has so many resources and so many people, not just physical resources but human resources. And we have a fantastic collection with specimens that we can use as comparative material. We've got a great collections team with X-ray facilities and photographic facilities. We've got a great library where we can look up all the references that we need. And we've got a fantastic genetics laboratory where we can run all the samples and create genetic trees.
Taxonomy, fish taxonomy research is sorely lacking in Africa. So the whole idea is to join forces, FAO with its research vessel and 32 partner countries, and SAIAB with the infrastructure, expertise and human resources to make SAIAB as a reference institute for building capacity in fish taxonomy, but also for carrying out fish taxonomy research.
So the immediate products are going to be research papers, which will help with the management of species within those countries. If you don't know what you have, you can't manage them in any way. So we're helping to identify the species that are present within those countries and revising checklists. In the longer term, though, we're giving scientists in those countries greater capacity. They're going to be able to do this themselves in future as they develop. So I think it's a very long-term impact that we're going to have.
Wow, it's been amazing. Amazing is the right, or spectacular, because I'm being surrounded by expert people who are in this world, in this field, work so long, and then they share the experience. They teach us, they teach me so many things, and then for me it's been amazing. This collaboration with the SAIAB with FAO is helping me a lot. To be in the lab learning how everything goes and the importance of all these kind of things, like the genetics in the end of the day was like beautiful, and how the morphology, the genetics complement each other, that we can't do taxonomy now without the genetics, you can't do the genetics without the taxonomy. The morphology part was amazing to be there, just to be explained how the tissue that we collect and the fish that we are made in like the morphologic, and then we have so many steps. So I love it, the morphologic, but I love also because I never been into genetic labs to do a sequence to know everything, how we get the sequence. I love it.
This is my first time actually to come to SAIAB. Before, when we were in the ship, the FAO taxonomic expert, Peter Somadakis, he told us that SAIAB, they have a very nice museum, and it will be very interesting if we could conduct our workshop there. So he said he will plan, FAO will plan to conduct the workshop in South Africa, Grahamstown at SAIAB. It is well organized, and we were, of course, taught so many things, including how to identify fish and how to follow the procedure for preserving the specimen, but also how to follow the procedure step by step so that you can reach a point to determine or to find that either new geographical record or to establish a new species. So this was very interesting. But apart from that, also we get an extra experience. They taught us, we have learned a lot about GIS, and also we also taught about molecular, how to do extraction, and also PCR analysis, so that you can understand the specimen you're getting. Also, apart from depending on morphometrics or classical identification of traditional method, we used it to have in taxonomy, but also molecular through barcoding is another additional input that we got from this training.